Okay, everyone. So let's go ahead and get started. So it's 2 o'clock, or close enough to 2 o'clock. So a couple of things just right off the bat in terms of the logistics of the exam. So the exam is going to open tomorrow at 8 o'clock. If you didn't know that already, and it's going to close at 6 p.m. You're going to have 80 minutes to take the exam. At 6 o'clock, the exam will turn off, even if you're not done with your exam. So you'll want to start your exam early. I would suggest no later than 4 o'clock tomorrow. Okay. The exam itself is going to take the format that is identical to the format and the formatting that you would see in the practice exam. It'll have essentially a matching question, several multiple true-false questions, and then finally it will have 3 short answer questions. Okay. And it's going to cover lectures 1 through 5, as you probably already hopefully know. So it will be, of course, remember, proctored by Respondus lockdown browser. I posted, an announcement about that on, I think it's Thursday of last week. And I also, in that announcement, mentioned that if you take this Respondus practice quiz, then you can get 2 points extra credit. Really, what I would like to see is that everyone does that so that we can handle any potential issues before the exam actually starts, right? So do that. If you haven't done that already, do that by tonight. Get yourself a small amount of extra credits, not a lot, okay? Essentially equivalent to 1 question on the exam though. And then hopefully those issues will be dealt with, okay, for you. Hopefully you don't have any issues, and hopefully you don't have any issues tomorrow. If you do, then we'll try and deal with those as they come up. If you do have issues, go ahead and email me, and then I will try and of course, quickly respond to your email. Worst case scenario, we'll have to contact Respondus web support, in a last ditch effort. If we just cannot figure it out, we'll figure something else out. Okay. All right. So I will work with people, and I will be available all day tomorrow to work with people on getting their exam taken. Okay. All right. So as far as this Q&A session goes, if you're just joining us, I have opened up this Poll Everywhere Q&A. I'm going to try and answer a question pop up here. So please go ahead and do that by all means. If you have like a question as I'm talking through these things, you can put it in the chat or you can put it there. I don't know if I'll get to all these questions. We'll try and get to all of them and through all of them. If you see a question, you're like, yeah, that's the one I want. And the more votes a question gets, the higher it gets on the priority list. And so I'll deal with those ones at the top first. Okay. You can also feel free to mic in and ask your question. I do not mind being interrupted at all. This is a fairly informal setting. I am recording it. So, if there's something that you're like, yeah, I don't know about that, you can go back and listen to that or something to that effect. Okay. Okay. So that's what we're going to do. So let's start answering some of these questions. I'm going to go ahead and turn off my video. That way I can use my tablet and write on my tablet and not have you staring at my ceiling when I put it down to write on it. So, okay. All right. So let's look here. The first one, are we allowed to, are we allowed blank scratch paper on the exam? Yes, I'm glad that you asked that. Yes, I would be willing to let you do that. And so I know that virtual exams are hard for students because there's nothing to write on. So go ahead and feel free to bring one of those to you or with you to your exam and write, down on those things. Obviously, it has to be blank. Okay, so this is not an open note exam. It is a closed note exam. So okay. All right, so let's look at this next question. This looks more like a content question. In gluconeogenesis, does converting pyruvate occur in the converting pyruvate into oxalic acetate occur in the mitochondria, or is it the pyruvate kinase just made in the mitochondria? So it's actually in the mitochondria, right? So if we were to do this, let me go ahead and I like to draw these just to give you a visual. So we take this and look at it from this standpoint. In terms of gluconeogenesis, what you would have here is pyruvate that will be out in essentially the cytosol. Okay, and then in here would be the matrix. Okay, and of course the source of that pyruvate would be something along the lines of amino acids. Okay, you can make that. Lactate can be used to make this. Okay, and then that pyruvate is going to slip through here, through the outer membrane, and then there would be some sort of transporter, a pyruvate transporter found in the membrane of the mitochondria that then the pyruvate would go through. Oops, wrong color pen. There we go. And get into here. The enzyme that then carries out the first step of gluconeogenesis is found really only in the matrix of the mitochondria. And it is going to convert that oxoacetate, I'm sorry, that pyruvate to oxoacetate, and you have to have a bicarbonate to do that. You're just going to add that. And you have to have ATP. This is an ATP dependent process. Okay. And that, again, that enzyme is pyruvate carboxylase. Now, the fate of that oxoacetate then, right, it can be converted either directly to PEP, or it can be converted to malate. And either one of them, okay? So if it's converted to malate, then the malate will be shipped out through the malate aspartate shuttle. If it's PEP, then that will be shipped out through a different process, okay? But nonetheless, that's what's going to happen here, right? So then, for instance, malate can then go through, let's have another here. Malate could come out here, and then that malate can be converted back to oxoacetate by malate dehydrogenase, and then that can be converted to PEP. Or this PEP can go through maybe another membrane transporter found here, and then that can be converted to PEP. Or not converted to PEP, but it'll just be shipped out. And then, of course, that is going to go on to make glucose. Or this is going to go on to make glucose. Okay. Now, what I'm not drawing here is the entire malate aspartate shuttle, but the entire malate aspartate shuttle will be actually used to do this. So, okay, all right. So hopefully that answers that question. Okay. We'll be given the Michaelis-Menten equation if we need it. If you need it, I'll give it to you. So, yeah, we give it to you. I'm expecting numbers up. Okay. We've already talked about that one. On the practice exam, I was confused whether the bold was true or false. So the bold is true. So on the practice exam, okay. We just talked about that one. In gluconeogenesis, does converting pyruvate into oxoacetate occur in the mitochondria, or is the pyruvate kinase just made in the mitochondria? Okay. Can we go over short answer problem 3 on the practice exam? Yes. So I pulled up the practice exam. And let's go ahead and look at that one. I think that this is the one that we want to look at. Yes, this is it. Okay, so short answer number 3. Let's look at this. Okay, so it's kind of spread out here. The binding of glucagon to its receptor rapidly activates the conversion of metabolite A to metabolite E through the pathway shown below. Awesome. Given this information, which of the following enzymes would you expect flux control coefficient C greater than 0.1 and explain why? List the ways in which these enzymes might be regulated. Okay, all right, so this is a hypothetical pathway. Let's go ahead and zoom in on this. Okay, and it's obviously this pathway conversion of A to E through these several intermediates B, C, and D. There's 4 different enzymes found there. And the question asks us to highlight which is going to have C values of greater than 0.1. Okay, a flux control coefficient values greater than 0.1, and then explain why. Okay, so when we look at this, okay, you've got 2 different types of reactions. I'm going to circle these ones here. Oops. Let me go ahead and circle the whole reaction for both of these, actually. You got this one here and this one here that have these delta G values that are, less than 0, negative, quite negative, actually. And then you've got these two here, this one, and then this one whose delta G's are 0, okay? These would be reversible type reactions. Okay, and those are going to be, right, these are going to have fast or high forward forward rates and reverse rates, actually. Okay, and they'll be what might be considered close to equilibrium. So if you were to think about it this way, right, so the VF of this, right, so if we were to draw it as this way, this is a forward, this is reverse, we would say that the rate of these, this could be, right, a VO equal to like 410 or something like that, just to make up a number. This one would be, VO equal to like 400. Okay, so the forward and reverse rates are almost identical to each other, and they're quite high. Okay, and that would be true of the other one. I won't draw that out. Okay, now when you look at those other reactions, right, these guys, these would be those irreversible steps. Okay, and these are going to have slow forward rates. And I would say this, even slower reverse rates. Okay, so if we were to look at this and do it this way, right, we could say that the rate of this, the VO of that, is going to be like equal to 10, something like that. Well, this, the VO, is going to be, a tenth of that, or something even less than that, is going to be like 0.5 or something to that effect. Okay, it's a very slow reverse rate, but in comparison, right, if we look at this to this, right, a whole order of magnitude less than what we see with the reversible rates, and they're very slow. Okay, and that would be true of the end one as well. Okay, and it's those choke points, we've talked about this in like one of the worksheets that we did, these rate limiting steps, the irreversible steps that are going to have the greatest C values. So we would say that the C values of these, right, would be, this would be a C that would be greater than 0.1. And this would be a C that would be greater than 0.1. Now, I wouldn't expect you to, right, say, okay, this one is 0.3 and this one's 0.7. There's nothing that we've done in this class that would give you the information to actually get a prediction of the actual value of the C, other than, that it's greater than the 0.1, meaning that these have the greatest contribution to flux through the pathway, okay? Now, having said that, if I said that this one is equal to 0.3, then there's only two of them, then you could say the other one is 0.7 because they do add up to 1, so I'd expect you to know that, okay? But nonetheless, okay, coming back to this, these are going to have the greater C values that have the greatest contribution to changing flux, okay? Now, in regards to, why, right? Because it then goes on to say this, explain why, and that's a very important part of the question of this, okay? Because I think that probably, the easiest portion of this is just to say, these are the ones with the largest C values. The question is, well, why is that the case? And this is why we spent some time on this in terms of like the worksheets, okay? And hopefully some of the things that we talked about in the worksheets made sense, but let me go ahead and draw this out, okay? And kind of just expand on that and go back to this idea. I like this analogy, I think it's a really good one, of bottlenecks, okay? Our highway, if you will, for this pathway is gonna look something like this. Okay, now I'm not going to ask you to draw anything like this on the exam because it's a virtual exam, so you have to explain with words. Okay, but we have A here and we have E exiting here, okay? And these 2 points here, here, and here would be our bottlenecks, they'd be a rate limiting step, okay? And when we think about then the capacity of this particular pathway to produce E, that capacity, when we think about capacity, really we're thinking about rate, is going to be most drastically changed when we change, okay, the slowest steps, this step and this step, right? If you think about it this way, we could redraw this way, and if we were to change these middle steps, you could see it something like this. Oops. Let me go ahead and erase this. You can see it something like this. If we were to change a middle step, we could go like this. Okay, if we did that, that's not going to do very much, right? Just increasing the rate through one of these already really rapid steps is not going to do much for the rate of the pathway, right? If you think about it in terms of cars getting through the interstate, we're not going to be able to do much because we're really, our rate limiting step is these choke points that we have to expand, okay? And so these really slow ones, these irreversible ones, these ones that have these negative delta G's, these are going to be the things that are rate limiting, and therefore we can increase rate of the entire pathway by increasing the rate limiting step by just removing the bottleneck, okay? Right. And then it says list the ways in which these enzymes might be regulated, also a very important part of this, okay? And we talked about several ways that this can happen, okay? And let me just go ahead and mention them. The first way is through either cooperation, right? A cooperative protein or an allosteric protein. Those are fairly common. In particular, allostery is a very, very common means of doing this. But the other ones would be covalent modification, and then finally change in genetic expression of that enzyme. Just have more of the enzyme available, and then that can change the flux through these pathways. So those are the 4 main mechanisms that we discussed, and also, quite honestly, the 4 main mechanisms that we will, as we move into metabolic regulation, that we'll see examples of. In particular, allostery and covalent modification are really, really common. So we'll talk about changes in genetic expression. We just won't look at it in a lot of detail. Okay, all right. So hopefully that helps you understand that question. Let me come back now to this. Make sure I didn't miss any. Okay, we got that one. Got that one. Got that one. Okay. Yep. Oh, so next short answer. So it says, can we go over short answer number 2? I think it just disappeared because it went to the top. Short answer question number 2 for the practice exam. Yes, we can do that as well. So let's look at that. And short answer number 1. Just do them all. So okay, so let's look at those. It's good that you guys are looking at the practice exam. So we'll do it in that order though. Number 2 and then number 1. All right. So the intracellular concentrations of glucose in the liver and muscle are 5 millimolar and 0.1 millimolar respectively. Okay, great. Describe the role that GLUT2 and GLUT4 play in maintaining such different intracellular concentrations for the 2 tissues. Okay, that's interesting. Okay, so what you see here is that the liver has a really high concentration of glucose in the cytosol relative to the muscle that has a fairly low concentration of glucose in the muscle cell, okay? And that would be relative to what you would find typically in the bloodstream, okay? So let's do this. So we've talked about several different aspects of these, and maybe we'll take some time to even expand on some of these concepts as we look into this. But let's first of all try and visualize this. And let's first of all visualize the liver membrane. Okay, so what we'll draw here is the liver membrane. And each of these boxes should have been the same size, but you know, I'm bad at it. Okay, because each of these boxes represents GLUT2 essentially. Okay, and we can go on and go with that. So this would be GLUT2. Okay, each one is GLUT2. Okay, now remember, Km of GLUT2 is really high. It's like 17 millimolar. It's super high. Okay, but essentially, when we look at GLUT2, because there's a lot of them in the membrane, okay, GLUT2 has the capacity, okay, and let's just say that this is the blood out here, and then this is, right, cytosol in here. Okay, and because there's a lot of them, right, one of the things that we see here is that, and let's use pink dots as glucose. Okay, but because there's a lot of them, these can actually equilibrate, okay, the concentrations of glucose on both sides of the membrane, blood and cytosol, right? So we would say this, that there is an equilibrium between these two, okay? So it would be, for instance, 5 millimolar here, and therefore it would also be, could be 5 millimolar here, okay? Now, essentially then what can happen is that, and as we've discussed in one of our worksheets, when the blood glucose concentration all of a sudden increases, okay, because these are at equilibrium with each other, then there'll be this pretty massive increase in concentration here from a percent standpoint. Okay, and we talked about the percent increase going from 4 millimolar to 8 millimolar. It was like 70% increase in that cell, right? And that liver, or this would be true for the pancreatic beta cell as well, they have a fairly massive influx change of glucose, and the physiology within those cells starts to change. And then they are going to take action, right? This would cause an increase in glycolytic flux, okay? An increase in ATP production. In the case of the liver, there would be other things that will start to happen. In the case of the pancreatic beta cell, that will lead to the release of insulin, okay? Right, so, but GLUT2 being, there's lots of it there, it's going to hold this thing in equilibrium, okay? All right, and then that glucose is going to go on and, you know, become something, right? So this glucose can become CO2 in water to produce, right, an ATP, or it can become, right, lipids. Okay, those would be the two major fates. Or it can become, there would be a third major fate here. You become glycogen. Glycogen, oops, sorry. Okay, so those would be the major fates of that, of glucose in that cell type. Okay, now here's the thing about the muscle. So now let's draw this. This would be liver or beta cell. Okay, although if you're looking in the beta cell, glycogen and lipids would not really be one of the main fates of that. It would be more along the lines of the liver. Okay, now when we look at the muscle cell, here's the interesting thing about the muscle cell, right? And let's do this. I'm going to draw this first of all in low blood glucose. Okay, the liver membrane is going to look like this. Maybe it would have like one of these or something like that, right? But it's going to not have a lot of GLUT4. It's just going to not have a lot of capacity to bring to bring glucose into the cell when glucose concentrations are low. In fact, what we would see here rather is something along this lines, that the GLUT4 is found in vesicles within the cell. Okay, so these will be GLUT4. Okay, and so you know, you don't, the liver, I'm sorry, the muscle cell. Okay, so this would be muscle. The muscle cell will not be at equilibrium in terms of concentration of glucose. So these little pink dots are glucose again. Would not be in equilibrium in terms of glucose concentrations on the outside of the cell and the inside of the cell. And the reason primarily being that there's just not a lot of transporter there when glucose concentrations are low in the bloodstream. Okay. Now, as we've discussed, okay, when glucose concentrations move from there to high, actually, let me draw it this way. I'll try and go down all the way down here. Oops. When we all of a sudden shift to high blood glucose, okay, this is going to lead to insulin production by the pancreas and insulin secretion, right? So now insulin's around. So what happens over here when we go from low blood glucose to high blood glucose, what happens over here in the pancreatic beta cells, a bunch of glucose comes into these cells, a lot more glucose is present in the cell, that starts to make more HCP, and that's going to lead to the secretion of the insulin, okay? Insulin then will bind to receptors on the muscle cell, and that's going to cause the mobilization of these vesicles to then go up and do this, right? Insert a whole bunch of GLUT4. Into the membrane, so that all of a sudden then okay, glucose will start to rapidly move through these transporters into the cell, and glucose concentrations will start to rise in this environment. Okay, now here's the interesting thing about all of this, right? And the question doesn't really ask you to state this, so maybe it was a poorly written question from this standpoint, but one of the things we know is going to happen, right, is this is glucose, right? Is that it's going to be converted to glucose 6-phosphate. That's the very first thing that's going to happen to this. This is going to go on and become glycogen in the muscle cell, or it's going to become really pyruvate, and then eventually CO2 plus water, or lactate, right? Lactate would be another fate in these cells. Okay, depending on if you're like, you know, working hard or something along those lines. Okay, so those are the fates of that glucose molecule. However, as more and more glucose comes in, this enzyme that's found here would be hexokinase 1 through 3. That would be the isoforms of this, right? Well, those isoforms are sensitive to feedback inhibition by glucose 6-phosphate. Okay. Right, so what happens here then is the reason that we have such a disequilibrium between the extracellular environment and intracellular environment, 5 millimolar out here, let's say this is, you know, 5 millimolar, and this would be 0.1 millimolar, and then we come up to here, this would be maybe 8 millimolar, and you would have a momentary increase, right? This would be up to like 0.5 to maybe even 1 millimolar. But then it would start to be brought down by these processes of making glycogen or starting to convert it to lactate and carbon dioxide and water. But what will happen though is that this glucose 6-phosphate will start to accumulate if the rates of these pathways, right, the rate of this or this or this is fairly low, and that will start to accumulate and then inhibit this, and then that will feed back because that's inhibited, okay, that will feed back and start to slow the progression of glucose into the cell, okay. So, this question only asks you to relate it GLUT2 and GLUT4, okay, and the biggest difference would be that GLUT2 is there all the time, okay, and GLUT4 is not. It's insulin sensitive. So, okay. All right, so hopefully that helps people out. Okay, now coming back to our where is it? There it is. Our questions here. Okay, so we just did 3. We've done 3. So let's do for short answer 1. There's a couple questions on this. How would the answer change if standard free energy was given instead of actual free energy? Okay. Short answer on practice 1. Could you briefly go over gap TH pathway? Yep. So okay, let's move on to question number 1 here. And look at this. Okay, so the ATP demand or ATP, the demand for ATP during strenuous anaerobic exercise increases drastically. Suppose skeletal muscle was devoid of a functional lactate dehydrogenase. Given this, predict what would happen to the concentration, increase, decrease, or stay the same during strenuous exercise in abnormal skeletal muscle relative to the resting of the following metabolites, and explain the rationale for your selection. Okay, awesome. So let's go ahead and look at this. Okay, so first of all, we need to start with this. Glucose, a rapid form of energy, right? And we convert it to glucose 6-phosphate, and then, right, this is converted to fructose 6-phosphate. That would be a reversible step. And then this would be converted to fructose 1,6-bisphosphate. And then this is broken into 2 molecules, G3P and dihydroxyacetone phosphate. This is converted to more G3P, converge on this, and we get to this, and that would be 2 1,3-bisphosphoglycerates. Okay, that one, I'm going to run out of room here, but that's okay. I'll just kind of shorten this up. That 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate, and then 2-phosphoglycerate, and then PEP. And this would be 2 of these, 2 of these, 2 of these, PEP, and then finally to 2-pyruvate. Right, so that's glycolysis, okay? And the big thing here will be, there's a couple of points to go on here. We have, of course, ATP being used here. And then we have ATP being used here. And then we have here ATP being produced. And then we have here, down here, same thing, 2 ATP produced. Right, and importantly for our question, there's going to be needed 2 NAD plus to do this. Okay, all right, now it says anaerobic conditions. Increases drastically, suppose skeletal muscle is devoid of functional LDH. Okay, anaerobic, here it is. All right, so understand that what's happening here is this: pyruvate is converted in anaerobic conditions in particular, but really in all conditions, but specifically anaerobic conditions, you convert to lactate. Okay, well, that lactate, okay, takes that NADH and recycles it back to NAD+. The enzyme that does this is that LDH. Okay, that's the enzyme. Now, what the question is saying here is this: well, what if LDH is gone, right? There's not a functional LDH. Well, what's going to happen is that, of course, then this step is not going to occur. And that means that this recycling step's not going to occur. Okay. Now that will then lead to a decrease in this, because all of a sudden NAD concentrations will go down. Right? So what will happen here? Um, so if you maybe, I think that maybe you didn't mute yourself, so you might want to mute yourself. So anyway, if you're to um, if you're to run out of NAD plus, this becomes an artificial choke point, okay, if you will. Meaning that this will start to become a bottleneck. And what happens here is glucose is converted to, right, these to G3P. G3P can't get through this any longer because all of a sudden there's this huge choke point. And so what's going to happen to this is that G3P is going to go up. So this would be G3P. So that's going to start to go up in response to this because it can't get through. There's nothing that can take the electrons from G3P to become 1,3-bisphosphoglycerate, okay? Now, at the same time, what that means is this, too, if you really start to consider what's happening here, okay? If you think about NADH, okay, well, the small amount of NAD plus that's in the cell will be converted to NADH. And then as that happens, this narrows and narrows and narrows. There's nothing recycling it. So what's going to happen to this is going to go up and this is going to remain high, all right? Because, you know, the exercise starts and then glucose is converted to glucose 6-phosphate, it goes all the way to G3P. There's a small amount of NAD plus in the cell. The NAD plus is converted to NADH. You start to run out of NAD plus, so this will go down. But the NADH has no way to recycle, so its levels go artificially high in this regard. Okay, NAD plus starts to go down. Okay, now of course ATP levels, okay, are going to go down as well. You're using ATP up here, but you're not converting it any longer down here, right? Because you can't really get to that payoff phase. Oops, sorry. So you really can't get to that payoff phase, and when you can't get to that payoff phase, then you're not going to get ATP. Not to mention also the fact that it says that you're exercising, right? So the whole reason you've increased flux through this pathway is because your muscle cells are running out of ATP, so they need to make ATP quickly, right? Well, they can't get to the payoff phase, or their degeneration phase because we're running out of NAD plus now, okay? And then of course lactate concentration. So ADP, by the way, will go up. Okay, because ATP is being used, ADP is going to go up, and then lactate, by the way, is going to go down because there's no LDH, right? So that's going to go down. So okay, all right. Now let's come back here. Let's see here. Not that. Let's come here because there was a question on this. It said for short answer 1, how would the answer change if the standard for energy change was given instead of actual for energy? Do I have the wrong set of questions? Because that one doesn't apply to the one I just did. So maybe, you know, if you ask that question, maybe, you know, Mike in and explain that. Maybe
yeah, I think I thought it was 1, but it's actually 3.
Oh,
that I was talking about.
Oh, okay. Yeah, let's go. I was starting to think to myself, did I pull up the wrong practice exam? Go to 3 here. Yeah, so you wouldn't, so good question. You wouldn't report standard free energy change for this. Because we don't, when you think about a question like this, we're thinking about the way the pathway is designed, and that is a biologically relevant situation, okay? And because standard free energy chains really are just artificial, right? It's a standard conditions, you don't report those because they have no real bearing on which of these is the actual choke point in the cell, okay? As we go through, and you may have seen this with glycolysis and gluconeogenesis and that kind of thing, there are standard free energy changes all over the map, okay? And they're usually quite a bit different than the actual free energy changes. And so really when we get to metabolic control in particular, and which of these are rate limiting and not, you're really only thinking about actual free energy. Okay, good question. I can see where that would come from. So, okay. All right. Could you briefly go over the gap DH pathway? Oh, yeah. I'm wondering if this is, let me ask this. I don't know who asked this question, but do you mean the glycerol 3-phosphate pathway? Is that the one you're talking about? Not the malate aspartate shuttle, not the lactate dehydrogenase, but the glycerol 3-phosphate shuttle. Is that what you're talking about? Which is
Yes.
yes. Okay, great. Right. I was wondering. So GAPDH is actually not the enzyme that's used in it. That's why I was trying to clarify. It's really similar. So GAPDH is glyceraldehyde 3-phosphate, but you have glycerol 3-phosphate is actually the metabolite found in this little shuttle. Okay, which is only it's an alcohol versus an aldehyde. Okay, so but anyway, so here let me think through this. Okay, so here's how this pathway will go. Okay, this pathway begins with, let's see here, how to best address this. You would do the same thing that we've talked about. So you would talk about the fact that G3P is converted to 1,3-bisphosphoglycerate. Okay, this would be part of glycolysis. Okay, that would be step 6, and that's going to require that NAD plus converted to NADH. Okay, so the malate aspartate shuttle is one way to convert that NADH back to NAD plus. The lactate dehydrogenase is one way to do this as well. The other way to do this is to do it through this glyceraldehyde or glycerol 3-phosphate pathway. So you would really start with dihydroxyacetone phosphate. I'm trying to remember. And then that is converted to this glycerol 3-phosphate. Okay. And the NADH, again, I'm going to drew this the wrong way, has to be used to do this. We're going to be doing it essentially like this, right? NADH is used to reduce the dihydroxyacetone to glycerol 3-phosphate. Okay, and then this will go on and be converted right back to dihydroxyacetone phosphate. And when that happens, there will be actually this FAD, okay, that's converted to FADH2. And then this is going to go on and do things. We'll just say things, okay, in the cell. It will really be in the matrix of the mitochondria. So if we were to put this all kind of in context here, do this. Okay, this would be the intermembrane space. This will be the cytosol. And then this actually takes place here. This would be, I'm going to actually draw the membrane. Okay, so these are meant to be lipids. Okay. And then we go like this. And this would be actually the inner membrane of the mitochondria. And this would be the matrix. Okay, that conversion that we're talking about here actually occurs within the membrane. Okay, there's an enzyme that's found in the membrane that does that. Okay, And the things, the reason I just say things, it's actually this coenzyme called QH, or it's actually coenzyme Q. And we're going to talk about that when we get to lecture 9 and what it does, and we'll come back and make this connection. Okay, but this is simply what happens here, and there's just this kind of recycling of NADH. Okay, it doesn't get much more complicated than that. We haven't talked about things yet to come. So, okay. Great. Okay, so, okay, could you go, we did that? Okay, how closely should we know the specific enzymes in the metabolic pathway? You know, so this is always a difficult question to answer. You have to be able to speak the language of the exam, right? So in other words, there will be questions that will reference GAPDH. There will be questions that reference PFK1. There'll be references to hexokinase and to GLUT transporters, and there'll be all sorts of these references. You know, a malate dehydrogenase, and all sorts of enzymes that we've talked about. And they'll be referenced in questions to direct you to particular pathways so that you know how to solve problems for those particular pathways. And so it's going to be important to be familiar enough with the pathways and the enzymes in the pathway to be able to speak the language of the exam to answer them the questions, right? There's not a question on the exam that says diagram this, like what you did for problem set 1 or even problem set 2, okay? There's not that kind of question where it's just straight memorization. I just have to write out the pathway. Rather, there's going to be questions that that you will have to know the pathway and how it connects to other things in the cell, that then, given the description of maybe some disruption of the pathway or increase in the flux through the pathway, that you will be able to think about how that would be connected to the other pathways and change rates and that kind of thing. So, okay, so that's kind of what you'd be looking for. So, you know, the way I've answered this in the past is, you know, I don't ever discourage memorization, but certainly don't stop there. Okay, if you've taken a physiology class, you know a lot. You can know a lot more about the physiology of the human body if you know the anatomy.
Move my shit. Oh God.
So someone's not muted, so you might want to mute yourself. So make sure you're muted. So, but anyway, so you know, the knowing the pathways, the actual and the enzymes in the pathways, kind of knowing the anatomy, so that then you can answer questions on the physiology, if you will. Okay, and the questions that are on the exams are going to be more related to like the physiology, the function of things. So, okay, all right. Let's come up here and make sure there's. We do this. Okay, short answer 2. We did that. Okay. We did this. We did that. We did that. We did that. Do we need to be able to differentiate between different types of GLUT transporters and their KN? And yeah, yes, I would. I mean, the ones that we talked about, in particular, GLUT2 and GLUT4 probably being very important ones. And then, you know, we talked a little bit about GLUT3 in the worksheet. So, so those would be the big ones that we spoke about. Dovagal pathway is upregulated in cancer cells. Can you talk about why that is? Actually, so I'm going to go ahead and wait on that. There's actually, we're going to have entire lectures devoted to that as we move on in the class. It's connected to lots of other aspects of cellular physiology, so we haven't really built the framework for that question yet, at least not in this class. So I want to build that framework, and then we can start to look at why glycolysis is so upregulated in the cell. Okay, and it's connected to the citric acid cycle. It's connected to also oxidative stress. And so I want to talk about those things before we get there. So great question. It's a very interesting thing, and we'll look at that as we go on. I think I just had something switched. Okay, museum. Will there be? Are we allowed on the practices? Used? Yeah, yep. So we got all these. Okay. Could you explain how mass action principle relates to flux? Oh yeah, that's an interesting question, and certainly one that is helpful here. So mass action principle. So let's go here and think about this. So when we think about mass action principles, what we mean is this, okay? Let's just draw a hypothetical pathway that has A goes to B, and we'll just say that that's reversible or irreversible, and then B goes to C, and then C goes to D, okay? And so what we have here, again, if you were to think about this metabolic pathway in terms of a highway, you draw it something like this, okay? So it looks something like that. And so when we think about this choke point here, this bottleneck here, okay, anything that gets through that is converted to B, and B goes up, and that automatically, because the rate of this, remember this is a reversible step, right? And because it's a reversible step, it has a really high forward rate. That shifts automatically towards C, and then C goes up, and that shifts automatically towards D, okay? And then D goes up. That's mass action principles is what we're talking about here, okay? These two steps here and here, okay, are at equilibrium, and so they respond very quickly in the direction that the stress is put on them, okay? Meaning that in this case, the stress is put on by the production of B, so they'll shift towards D, C to D. Okay, now there could be some alternate pathway that has X that's converted to D, okay? And there would be some regulatory point here, and when X is converted to D, well, then that's going to cause D to go up, okay? And this is going to go in the opposite direction, right? Now you say, well, there's going to be a collision there. Well, here's what's really important about this. When you have two opposing pathways, if this here, this step here is upregulated, then this step here at the beginning will be downregulated, okay? So that you'd have production of D that would then shift the entire thing in this way, and you wouldn't have really much production of B. Now this would have to, you know, maybe if you're going in like something like gluconeogenesis, maybe there's another enzyme that converts B to A, okay? And of course, the opposite would be true if, right, you're producing B, flux, cellular regulation signals are increasing flux for this, then they would be decreasing flux for this. There would always be some reciprocal regulation of these. And that will be something that we really highlight in particular as we get to glucose regulation. Okay, we'll be thinking about this and thinking about how glycolysis and gluconeogenesis are reciprocally regulated with respect to one another. So, okay. But mass action, going back to that mass action, is just essentially this idea of whatever is being produced, okay, is going to put a stress on these at equilibrium or reversible reactions, and they're going to respond in the direction opposite of that stress, okay? So B goes to D, or D goes back to D, depending on which stress is being put on, okay? Hopefully that answers your question. Okay. Okay, so are the Km values going to be given, or are they required knowledge? So again, you're not going to get points taken off for, you know, GLUT2 is got a Km of 17. So, and GLUT4 has 1 of 5, and GLUT3 has 1 of 1. But I would expect you to know that GLUT2 is high, okay, really high, and that GLUT3 would be really low. Okay, but I'm not going to ask you to state the explicit number. Okay, the glycolytic pathways upregulating. Okay, we already did this. We need to know that. Okay, so I think we got all those. Sorry, I'm getting lots of messages here. How closely should we know? Okay, I did that. Can you briefly go over the graph you created to describe the difference between Q and KEQ? Yeah, let's go ahead and do that now. And that will be, I think, probably the last question. Just a second. Look at these other ones. Gluconeogenesis is exclusive to the mitochondria. Pyruvic carboxylase is, but gluconeogenesis takes place in 3 different compartments in the cell: the cytosol, the mitochondria, and also in the ER. So How specific must we understand the metabolism of the pathways? Must understand. I think we've already talked a little bit about that. We have scratch paper. We've talked about that. So yeah. So exact numbers, no. You're not going to need to know exact numbers. What is the format? Okay. So we already talked about the format as well at the beginning. Okay. So let's come back to this. Okay. And look at this. So this graph that we talk about here, I believe that the question is in reference to this. Let me make a better version of that graph, actually. There we go. Call it free energy landscape. And you're plotting G essentially versus a reactant product ratio. Okay, so essentially what you have on your x-axis is what would be called Q values, or these are, right, concentration of product to concentration of reactants. Okay, and I like to, when I draw out a graph like this, I like to give it some, you know, landmark, which is 1 to 1, around there. And then what you do here when you look at this, okay, the graph will look something like this, usually. Okay, or it could be, I mean, there's lots of different, obviously, versions of the graphs depending on what your delta G values are and that kind of thing. But essentially what we're doing is mapping out how free energy changes as a function of changing product to reactant ratios. Okay, and usually, not usually, always at the low point of the graph, this point right here, that product reactant ratio, the very specific one, it would be the KEQ. Okay, because the KEQ by definition is where the free energy of a system has been minimized. Okay, what we're showing here is free energy as a function of product reactant ratios. Okay, and so at the minimal point of this, we're saying that we're at the equilibrium value, or the minimum point of free energy. Okay, what might be considered the ending point of the reaction. Okay, all other points along this, okay, are going to be Q values, different starting points. For instance, we could start all the way up here. Okay, this would be some Q value that would be an actual condition. That's the product to reactant ratio, let's say, of ATP and ADP in the cell. Right. Well, what you see here is this, that it's a long way removed from its equilibrium point, meaning that it has a high potential energy or chemical potential to drop to lower free energy values as it goes towards equilibrium. Right, and because this reaction is going to go towards products, right? We have mostly reactants over here, mostly products over here, right? It's getting to larger and larger product values. Because it goes towards products to reach equilibrium, this would be like an exergonic process. Free energy is coming out of the system. Okay, it's spontaneous. It produces products to reach equilibrium. Okay, right. And the reason I like the graph is because it kind of tells me, okay, you know, the closer if I have this as, like, let's say Q2, the closer I get to my equilibrium value, of course, right, the smaller the free energy change that I go, right? So if I were to say this would be Q1 here, that would be Q2, right? With Q1, okay, this would have a delta G that would be like negative 25 kilojoules per mole. And if I were to look at Q2, because it's closer to equilibrium, if I were to start there, that would be a delta G that would be maybe somewhere around negative 10 kilojoules per mole or something like that. Okay, the magnitude is a lot smaller. It's closer to its equilibrium value. Okay, right. So that's the graph that I like to use to try and explain actual free energy changes. So and essentially Q is the starting point for that particular process. Okay. Now, if you were to look at it this way, let me go one more step. Right, Q3. Well, this is a reaction whose actual starting point is more towards product than is reactants. It's going to have to go backwards to reach equilibrium. I mean, it's going to go towards reactants, and that delta G, so Q3, would have a delta G that would be like positive 5 kilojoules per mole or something like that. It would be positive because it's going backwards to reach equilibrium. So, okay. All right. So it's 3 o'clock, which means I need to to kind of get going here. Okay. I hope that this has been helpful. It looks like we got through many, if not all of the questions. Certainly, if you have questions, you can email me or you can hang out for a little bit. I might have a little bit more time to hang out, but I do have other things I got to do. But if you have one or two more questions and want to hang out, that's fine. But I'm going to go ahead and stop the recording at this point in time and let you all go. If you have issues as you go through the exam or trying to get into the exam tomorrow, let me know. Okay, go ahead and email me. Okay. All right. Take care, everyone.
Thank you.
You're welcome.
Thank you.
Absolutely.